Hi everyone. So today we are going to discuss on polymerase chain reaction, that is PCR. So PCR, wherein amplification of sequence of DNA occurs, and components required for this kind of instrument is DNA polymerase, which is an enzyme such as tag polymerase. Mm, Tag polymerase is derived from bacteria which survives at very high temperature. Example, Thermophilus aquaticus. So these specific enzymes have the efficiency to perform its function even at very high temperature. So second is primers. So primers are the short complementary sequence of desired gene act as the initiator in the PCR. And third is deoxynucleotide triphosphate. Consists of four different kind of nucleotides, that is four different kind of nucleotides, that is adenine, guanine, cytosine, and thymine. So what are the steps in PCR? PCR have three main steps. That is, first step is denaturation, wherein double-stranded DNA becomes single-stranded DNA. Second is annealing, wherein the primers attached to the 3' region of the single-stranded nucleotides at the 3' region, and initiates the extension where DNA polymerase will bind and starts recruiting complementary nucleotides on the 5' region of the primer, and extension occurs. So what are the things that should be kept in mind when you are designing a primer? So the length of the primer should be 18 to 24 base pairs, so that it will be okay. Then 40 to 60% of GC content should be there, and start and end of the primer should be with at least 1 to 2 guanine and cytosine pairs, as these form stronger bonds, double hydrogen bonds, compared to adenine and thymine. And melting temperature should be 50 to 60 degrees Celsius. Primer pair should be with a temperature difference of maximum 5 degrees Celsius. So forward and reverse primer should not have differences of temperature more than 5 degrees Celsius, or else non-specific binding will occur. And primer pair should not have complementary regions, because if forward and reverse primers have complementary regions, they will bind with each other and forms dimer, which will later obstructs or fails to amplify the specific gene that is required for to amplify. So what happens in general? In general, what happens is you have sequence of DNA or nucleotide, can be human DNA or plasmid or anything. And when you add primers and all the components required for the PCR, denaturation occurs at 95 degrees Celsius. Both the double-stranded nucleotides will form single-stranded nucleotides, and primer will attach to the 3' region of a specific complementary region, and which occurs at 55 to 65 degrees Celsius, and extension occurs. So this process will be repeated in next step. So in first step, when you have 2 DNAs, you will get 4. When you have 4, you will get 8, and 8 copies, and this cycle will go till 20 to 30 cycles. So what are the components required for PCR instrument? So DNA sample can be plasmid sample, human DNA sample, and primers, that is forward primer and reverse primer, and nucleotides, that is cytosine, guanine, thymine, adenine. Tag polymerase functions as addition of complementary nucleotides to the 5' regions of the primer. Mix buffer, which maintains the normal pH, and all these things are added in the PCR tube and run in the PCR instrument. So what PCR is used for? So PCR can be used in a medicine as a diagnostic kit, It can be used as a phylogenetic tree to study the evolutionary closeness. It can be used in forensic science, wherein we can use this as a DNA fingerprinting, and in food and agriculture, wherein we can make genomic modified organisms or plant, GMO plant, and consumer genomics, and environmental microbiology. So you can use it as a recombinant technology or anything. So thank you for your patience hearing, and please do not forget to like and subscribe my channel. Thank you.